Okay, a cell duplicate it is genomes. Okay, then the second one is that synthesizes the other constituent of the cell. Synthesizes the other constituents of cell. Okay, of cell. Synthesizes the other constituent of the cell and divides eventually into two daughter cells. And
So the right side board is not visible.
And divides eventually.
Sir,
Yes.
Sir, the right side
Yes.
board is not visible.
Okay, I'm just writing there. Okay, you see that is cell cycle is a sequence of events. I'm writing cell cycle is a sequence of events. Cell cycle is the sequence of events. Okay, it's the sequence of events. Cell cycle is the sequence of events by which. Okay, by which. What is cell cycle? Cell cycle is the sequence of events by which a cell duplicates it is genome. Okay, it duplicate it is genome. It is genomes. Then what it does? A cell then synthesizes the other constituent of the cell. Synthesizes the other constituent of the cell. Okay, that is constituent of the cell. That is little bit I'm doing that. Okay, so that you see that is synthesizes the other constituent of the cell and then divides eventually into two daughter cells. So what is cell cycle? Cell cycle is the sequence of events. There is a sequence of events by which a cell duplicate its genomes. Okay, that means how a cell duplicate DNA. That means how the DNA that is duplicate, synthesis of DNA, then synthesizes the other constituent of the cell. Synthesizes the other constituent of the cell and divides eventually into two daughter cells. That means a cell divides into two daughter cells. So what is cell cycle? It is the sequence of events by which a cell duplicate its genome, synthesizes the other constituent of the cell. Which are the other constituent? Cell organelles, different cell organelle, mitochondria, Golgi bodies, okay, endoplasmic reticulums. These are the constituent of the cell that are also synthesized, then divides eventually into two daughter cells. Okay, divides into two daughter cells. That is the cell cycle. What is cell cycle? It is the sequence of events by which a cell duplicates its genome, synthesizes the other constituent of the cell, and divides eventually into two daughter cells. Okay, that is the cell cycle. That is the definition of the cell cycle. And that means you see that. That means all the events of the cell cycle, you see that all the events of cell cycle, it is the sequence of events. All the events of cell cycles are under the control of gene. Okay, are under the control of genes. All the sequence of cell cycle are under the control of that of gene. Okay, that is genetic control. All sequence of cell cycle are under the control, are under the genetic control. Okay, if cell cycle or cell division are not under the genetic control, then what will happen if such cells, if divisions are not under the genetic control? Okay, if there are abnormal growth of cells, cell divisions are not controlled. That is, in such case, it will lead to cancer. Okay, it will lead to cancer. So if it is not under the, if cell cycle is not under the genetic control, cell division is not under the control of gene, then it will lead to cancer. Okay, it will lead to cancer. So you see that 10 to the power, how many cells in human being, in a matured human beings, there are 10 to the power 14 cells. And each cell carry oncogene. Each cell carry oncogene. Okay, each cell carries oncogene. And that oncogenes are not in, that is, they are not, that means when such oncogenes, that means they are at premature stage, oncogenes can have to be, okay, matured if radiation or some type of mutation occurs in our body, and if such oncogenes are not controlled, okay, such oncogenes are not controlled, or if cell division, if said the process of cell division does not come under the control of gene, then such oncogenes will be that they will have to be developed, they will have to develop and lead to cancer in the body. So that the process, the events of cell cycle are under, that means under the control, under the genetic control, gene control the cell cycles. Okay, so there is a systematic, okay, there is a smooth division of cells. So there is, cells don't divide randomly. Randomly division of cell will lead to cancer. Okay, so that there is a proper division, there is a proper channel, proper way by which cell divides, and that sequence of cell division is known as that of cell cycle. Okay, cell cycle, and that cell cycle is of two, that cell cycle includes to that of two phase. One is that of interphase, another one is known as that of that is mitotic phase. Okay, interphase and mitotic phase, and that cell cycle also, that cell cycle, it is that Howard and Pilley. It is that of Howard and Pilley. That means who? That means who for the first time? Who for the first time described? That is Howard and Pilley. That is Howard and Pilley. Okay, that means they describe the cell cycle for the first time in 1953. In 1953. Howard and Pilley, that means described the process of cell cycle for the first time in 1953. From 1953 onwards, that biologists know about that of cell cycle, and the bio students are knowing about that of cell cycle. Before 1953, there was no idea of such cell cycle developed inside our body. Okay, so that this Howard and Pilley, that you have to remember. That means he for the first time described about that of a cell cycle. Okay, and another thing, student, that mode of cell cycle is fundamentally similar. The mode of cell cycle and that of cell division are fundamentally similar in all organisms, which shows the unit of life. The unit of life, how you will know that there is a unit of life? That means the unit of life, that means is based on that of common parameters showing in that of cell cycle and cell divisions. That means cell cycle and cell divisions common in all organisms, showing to that region there is a unit of life. Okay, and then we'll have to know about that of all, that means that is generation time. That means time of cell cycle, that is called generation time. We'll have to know about that of okay, generation time. You'll have to know about that of generation time. What is generation time? The time between two cell division. Okay, period between two successive cell division. What is generation time? Period between period between two successive cell division. Okay, cell divisions. That is called generation time. What is generation time? Period between two successive cell division is known as generation time. And then cell cycle is not. That is cell cycle found in all organisms. But another thing is that cell cycle, that is generation time is not uniform in all organisms. That means that generation time varies from organism to organism. That is varies from species to species. Okay, so you'll have to know about that of some organisms, and that means it depends upon that generation time depends upon that means generation time is not uniform in all organisms, and it varies from organisms to organisms, and generation time depends on external factor, so generation time depends on external factors. That means temperature, food, oxygen supplies. Okay, that is temperature, food, and O2 supply. OT supply. That is the external factor, as well as the type of cell, type of cells. That means whether it is a prokaryotic cell or eukaryotic cell, plant cell or animal cell. So that it depends. So generation time found in all organisms, but generation time varies from organism to organism. Generation time varies from organism to organism. Why it varies from organism to organism? Because generation time depends on external factor such as temperature, food, and oxygen supply, as well as type of cells, whether it is a plant cell or animal cells, prokaryotic cell or eukaryotic cell. So it depends. That means prokaryotic cell generation time, eukaryotic cell generation time. Is it possible that both will be same? No. And that of plant cells generation time, animal cell generation. So that will also be varied. So that generation time that varies from organisms, and for such variation depends on external factor such as temperature, food, and O2 supply, as well as type of cells. Okay, type of cells. Then we'll have to know that you see that bacterial cells. We'll have to know that generation time of bacterial cells. Okay, generation time. That means type of cells. You see that type of cells and the generation time. I'm just showing you that of bacterial cells. Okay, bacterial cells. That is bacterial cells. That cell cycles. The cell cycles continue for that of 20 minutes only. Only 20 minutes. That of bacterial cells. Bacterial cells generation time is just 20 minutes. Only 20 minutes. So that is one cell division. After 20 minutes, there will be another cell division. Okay, and for that of yeast cells. Okay, for that of yeast cells, that is yeast cells, it is for that of 90 minutes. It is for that of 90 minutes. For yeast cells, it is for that of 90 minutes. That means one yeast cell will divide, and after 90 minutes, another cell will be divided. So there will be a gap of 90 minutes in between two cell divisions. And in case of that of epithelial cells of intestinal lining, so epithelial cells, that is epithelial cells of intestine. Okay, epithelial cells of intestines, that it will take 8 to 10 hours. 8 to 10 hours. Okay, it will take 8 to 10 hours. Epithelial cells. Then that of onion root tip cells. Onion root tip cells, and it is that of 20. Okay, that is 20 hours. That is, you must take that of 20 hours. And the human cells divide, that is 24 hours. Okay, so 24 hours. So there is a 1 day, it takes generally 1 day. One cell divide, and there is a cell division after 24 hours, that is and cell division. So that in between of that of two cell division, there is a gap of 24 hours. So type of cells, bacterial cells, 20 minutes. So very fast. And each cell 90 minutes. Epithelial cells of intestine 8 to 10 hours. Onion root tip cells 20 hours, and human cells 24 hours. So you see that generation time varies from species to species. And why that is such variation occur? It is due to that of external factor like that of temperature, food, and oxygen requirement of oxygen, as well as the shape of the that is shells. Okay, that bacterial cells or that yeast cells, bacterial cells 20 minutes, yeast cells rather develops, so it takes that of 90 minutes, and that epithelial cells 8 to 10 hours, and onion root tip cells 20 hours, and that human cell that is 24 hours. So there is a variation in that of a generation type, that is type of cell and a generation type. Okay, then we'll have to know about that of phases of cell cycle. Okay, phases of cell cycle. Suppose you have to see that phases of cell cycle in a cell. You see that. That is, just you mark it that of okay. Then you see that here is that. You see that here is that of here is that is M phase. This one is that of M phase. This one is G0 phase. This one is that of G1 phase. Okay, G1 phase, and this one is that of a little bit. Okay, G1 phase. This one is that of S phase. This one is that of G2 phase. Okay, and this G1 to that of G1 to that of G2 phase. G2 phase. This is called interphase. Okay, this is called interphase. And this is M phase, that is mitotic phase, and that mitotic phase also you'll have to see that is, first of all you will have to see that there is a prophase. Okay, that is prophase. Then we will see that is metaphase. Then this one is that is anaphase. This one is that of telophase. Okay, that is telophase. And this one is cytokinesis. Okay, that is cytokinesis. And this is karyokinesis. That is cell division start with karyokinesis. That is division of nucleus. After division of nucleus, that is division of cytoplasm. Okay, that is division cytokinesis. So that is this prophase, metaphase, anaphase, telophase. These are called that of karyokinesis. But after karyokinesis, then there is a cytokinesis. That is cytoplasma. Okay, and so that then that is G0 phase. G0 phase, that means after N phase, when cell will come to the G1 phase, it will decide that whether it will go to that of S phase or remain in the G0 phase. G0 phase is that of quiescent phase. That means it is a phase where there is metabolic activities is going on, but it is some sort of resting phase. Okay, that is rest phase. It is some sort of rest phase because if some cells are not required for any division, they will remain in that of G0 phase. There are some cells, you see that nerve cells. After differentiation, no, because nerve cells will not divide. It will only die and go, worn out. Okay, so that there is no middle division of that of neural cell. So they will remain in that of G0 phase. G0 phase. And there are some cells also that of heart cells. They will also, after differentiation, they will on that of G0 phase, because they only die. That is, they only have to die, but they will not divide again. And there are some cells also, that is fibroblast cells, which are requiring for the healing of wounds. That means such a division is not required. Fibroblast cells divisions, continuous division is not required. Whenever necessary, whenever there will be some wounds will occur in our body, then fibroblast cells are required for healing of that wounds. So for that, for the remaining period, when they have no one, they will remain in that of G0 phase. Okay, they will remain in that of G0 phase. So then G1 phase, that is G1 phase. G1 phase is the most important phase, because so many organelles also develop in that phase, that is G1 phase. And after G1 phase, then there will be S phase. S phase means that is synthesis phase. In this synthesis phase, there will be, that is DNA will be synthesized. Okay, DNA will be synthesized. And after S phase, that is G2 phase. In that G2 phase, it is a preparatory phase in which cells will have to be prepared for that of division, in which centrioles will be synthesized, and also that protein will be synthesized. I'll have to tell you. Okay, so that there is a G1 phase, S phase, G2 phase, and a M phase. There is a sequence. What is the sequence of that of cell cycle? First one is that of gap one, G1. Okay, G1. And then there is S phase, that is synthesis phase. Then we'll come to that of G2 phase. Then we'll come to that of that is M phase. Okay, that is M phase. That is G1, S, G2, M phase. This G1, S, G2, these are called that of interphase. This one is called that of interphase. And M is called that of mitotic phase. Okay, mitotic phase. Actual cell divisions, so that actual cell division occur in that of M phase. It is the most active phase. And the inactive phase is that of interphase. Interphase is some sort of inactive phase. But mitotic phase is that of that is mitotic phase is that of that is active phase. Most active phase is M phase. Most active phase. This one is the most active phase. And the interphase is that of inactive phase. Okay, inactive phase. That is interphase. But that is inactive phase. But growth continues. Okay, growth will continue, and that synthesis, DNA synthesis will continue, centriole synthesis, histone protein synthesis, all will continue. All will continue, but inactive phase means they will not interfere in that of cell division. Cell division is meant for that of M phase. Okay, M phase. And if you have to take that of 24 hours for cell division, M phase share is just 1 hour. M phase just take 1 hour for that of cell division. And if you have to come to that of G1 phase, G1 phase generally 8 to 12 hours. Okay, that is 8 to 12 hours. G1 phase takes 8 to 12 hours. Then come to that of S phase, synthesis phase. Synthesis phase generally take 6 to 8 hours. 6 to 8 hours. Okay, and the G2 phase, that is suppressed phase, that is 3 to 5 hours. 5 hours. If you have to see that 12 hours 8, that is 12 8, that is 20, and the 20 you add to that of 3, that is 23 hours. Okay, then if you have to add 12 6, 18 plus 5, that is also 23 hours. So interphase takes 23 hours, whereas M phase just take 1 hour. So cell division is the shortest affairs and the shortest period, and that is only 1 hour. For preparation, as you see that you are preparing NEET, that is you are preparing for NEET means you are preparing for 2 years. That is 40, that is 24 months you are preparing. You are preparing for 24 months. You see that you are preparing for 23 months. And you need that of month of May. Generally need to, due to that of COVID case, that means for 2 years that NEET examination is conducted in that month of September, but it is generally conducted in the month of May. Okay, so if you have to pick your preparation from that of May to that of, suppose you prepare from May to that of, or you start preparation from that of April. Okay, April, and to that of up to that of March. That means that is 221. Suppose you take, and you will have to appear the examination in 223. So that you have taken that of 23 months for the preparation. But 1 month, that is 1 month means only 3 hours. 3 hours. Okay, that is 23 months into that of 24 hours. That means what hours? That means you have taken from that of preparation of NEET examination. And only 3 hours. You have to appear that of 180 questions. You have to give that of answer of 180 questions. That will determine your distance and determine your chance in that of any medical college. So that similarly, interface takes that of maximum times for what? For preparation. If preparation is good, you can achieve success. If preparation is bad, how will you have to achieve success? So you see that cells takes 23 hours for preparation and just 1 hour for cell division. And cell divisions go, that is on a smooth basis. It is going to that of 23 hours of preparation. Okay, preparation. So you see that for that of a cell, for a cell cycle, that is G1 phase takes 8 to 12 hours. It is the longest phase, but it depends upon that of. That means if it is a frequently dividing cell, that G1 will take the less time. If it is a, that means if a cell is frequently divided, then G1 will have the less time. But if it is not frequently dividing cell, G1 will be the maximum time. That concept should be clear. If for the frequently dividing cells, G1 will be shorter. For not frequently dividing cells, G1 will be, that is, longer. That concept should be clear. Okay, and then that is synthesis. That is the DNA synthesis phase. It takes to 6 to 8 hours. It just takes 6 to 8 hours. Okay, DNA is synthesized here. That is histone protein also synthesized here. Centriole also, duplication of centriole also takes place here. That is synthesis. And in that case, that is the G2 phase. G2 phase, that is 3 to 5 hours. In this, protein is synthesized and it just takes 3 to 5 hours. So if you have to, that means on the basis of time taken, if you have to put in a sequence, then first will come that of G1 phase will take maximum time, then S phase, synthesis phase, then G2 phase, then M phase. Okay. So G1 phase, among the interphase, G1 phase, that takes the maximum time. Then S phase, synthesis phase, then G2 phase. And for that of N phase, that is, it is the shortest phase. Shortest phase. In a cell cycle, G1 is the first, it takes maximum time. After that, S phase, then G2 phase, and N phase is the shortest one. Shortest one. And among the interphase, G1 is the longest, that is phase, and that it is that of shortest phase. Okay, so you have to remember like this way. So that is if you have to take the human cell division for that of 24 hours, that M phase takes how many hours? That students, M phase
1 hour.
1 hour, and G1 phase
8 to 12 hours.
8 to 12 hours, and then come to that of synthesis phase
6 to 8
6
hours.
to 8 hours.
hours.
6 to 8 hours. And that of G2 phase?
3
that
to
is
5 hours.
yes, 3 to 5 hours. G stands for
Sir,
gap, gap. Okay, gap. And A stands for
synthesis.
Synthesis.
synthesis. Okay. That means what? That is what is synthesized in this phase. That is a DNA. Okay, DNA. It is the only phase. That is the only phase where DNA is synthesized. Okay, DNA is synthesized. You see that. Suppose in G1 phase, that is 2C. The amount of DNA is 2C. And after the end of S phase, it will be that of 4C. Okay, 4C. You see that if the amount of that is DNA in G1 phase, it is 2C. That amount after synthesis phase, because in synthesis phase the amount of DNA becomes doubles. Okay, doubles. So the amount of DNA after S phase will be 4C. And in M phase it will divide, so that it will be 2C. Okay, it will be 2C. Cell will be divided. One cell will be 2-1. So in one cell it will be 2C, another cell it will be 2C. And that cell come to that is 2C. And in this phase 2C will become 2C. 2C will become 4C. 2C, it is 2C. And whenever that synthesis period is over, then it will be 4C. And in that of G2 phase, it will be also 4C. But chromosome number will remain same. Chromosome number. Suppose chromosome number here is that of 2N. Here also 2N. Here also 2N. Here also that of chromosome number, it will be that of division, so that will be NN number. So chromosome number will remain same in all the stages of that of interphase. In all stages of interphase, chromosome number will remain same. But the amount of DNA, okay, but the amount of DNA will be doubled in that of synthesis phase. That is 4C. Okay, that will be 4C. And students, that in that today is that Friday, so in the evening class we will have to continue the cell cycle, and I will have to finish this topic of that of cell cycle in the evening sessions. Okay, so that now I'm just asking you only one question, so that conceptually, and you have to understand it, just answer the one question. That is very important, and you have to answer it. Okay, so students, question is that how much duration? Okay, how much duration? You write down the question. Okay, how much duration? Question is that how much duration is required? How much duration is required? How much duration is required for the formation of 8 cells? For the formation of 8 cells. 8 cells in yeast. 8 cells from a single yeast cell. From a single yeast cell. Okay. How much duration is required for the formation of 8 cells from a single yeast cell? Students, can you answer? Can you answer it? I'm giving you option. Okay, I'm giving you option. That is 90 minutes. B, 180 minutes. C, 270 minutes. And D, 360 minutes. Have you any ideas? Anamika A, wrong answer. Smaranika Patra, right answer. Smaranika Patra is right answer. Beula wrong, Jyoti wrong, Beula wrong, Tushar Ranjan right answer, Tushar Ranjan Meher right answer. Okay, you see that students, I am just that Bhagavat Hembram the right answer, Biswajit Mahalik right answer, Abhilip Sapal right. Okay, you see that. You see that. I'm just showing Pradimna Sahu wrong answer. Pradimna, you see that. I'm just solving such question. Okay, you see that how much duration is required for the formation of 8 cell from a single yeast shell. Yeast shell for a single division. So that is time. That time 90 minutes. Okay, a yeast shell takes 90 minutes for cell division. So that there is a gap of 90 minutes in between the yeast shells. So you see that one yeast shell divides into that of two shells. Okay, two shells, that is in 90 minutes. That is 90 minutes. The two shells divide into that of four cells. Four cells, that is also 90 minutes. Four cells also divides into that of 8 cells. That is 90 minutes. So 90, 90, 90. That is 270 minutes. Okay, 270 minutes. We'll have to see that. 8 is equal to 2 to the power 3. So that one cell division requires what? One cell division requires 90 minutes. So there will be 3 cell division. So 90 into 3, that is 270 minutes. So right answer is 270 minutes. Swarnika Patra has given the right answer. Okay, Swarnika Patra, you are not sending your back assignment to very bad indeed. Okay, so that you see that. So that how much duration is required for the formation of 8 cell from a single yeast shell? Answer is 278. You will have to convert 2 into 2 to the power 3. That means there will be 3 cell division. There will be say 3 cell division. One cell division requires 90 minutes. So 3 cell division will require 90 into 3, that is 270 minutes. So answer is 270 minutes. Thank you, students. In the evening sessions, so there is a communication gap owing to that there is some sort of problem today. So that in evening session, I will have to complete this portion, this topic of cell cycle. Thank you, students. Thank you
Thank
very
you, sir.
much.
Thank you, sir.
Thank you, sir.
Thank you.
Thank you, sir.
Thank you, sir.
Thank you. Thank you.